Now let us discuss about the phagocytosis. Now phagocytosis is a way our body cell they engulf and destroy the harmful microorganism in order to keep us healthy. So remember that the phagocytosis it starts when the specialized cells in our immune system like the macrophages recognize and attach to the harmful microorganism using their surface receptors. So these receptors, remember these receptors can be mannose receptor or they can be scavengers receptors. So in phagocytosis, the first step is opsonization. So remember, the process starts with when the microorganism they are coated with special proteins that are known as opsonine, and this coating is known as opsonization. And this opsonization it will help the phagocytic cells bind more easily to the microorganism. So what are the main opsonine and their receptors? The first is remember it's the IgG. It is a type of antibody that is found in the blood. So it coats the bacteria and make them easy for phagocytes to recognize. So remember phagocytes they have receptors especially for IgG. Other is C3b opsonine. So it's a fragment that is generated by the complement system. So what it does is it will attract the phagocytes to the bacteria. And phagocytes they have receptor for C3b2. Other is lecithin. They are the carbohydrate binding proteins and they bind to bacterial. So this one is IgG receptors. This one is the Fc receptors. This one is lecithin. So this one is the opsonization stage. So can anyone tell me why the opsonization is important for phagocytosis? So remember, it makes it easier for phagocytotic cells to bind and engulf the microorganism. So the next stage is engulfment. So once the microorganism is opsonized, the phagocyte is ready to get engulfed. So the phagocytes, remember, it will extend its membrane around the microorganism. So it will form a pseudopod. So it will form a pseudopod. Remember this pseudopod. It wraps around the microorganism, forming what we call is as a pseudopod. So these pseudopod would wrap around the microorganism, engulfing it in a bubble that is known as phagocytotic vacuole or phagosome. Now, why do you think the phagosome need to fuse with the lysosome? The lysosome, it contains digestive enzymes that break down the engulfed microorganism. So now the microorganism, remember, it is trapped inside the phagolysosome where it will get killed and it will get digested. So remember, this process, it can occur through the various mechanisms. One is the intracellular mechanism, that is the oxidative mechanism. It can be myeloperoxidase dependent, or it can be MPO independent. So the MPO dependent, that would use myeloperoxidase to produce toxic substance, whereas MPO independent uses other oxygen free radicals to kill the microorganisms. Other can be extracellular mechanism. Sometimes the killing and degradation, it can occur outside the phagocytic cells using some enzymes and the toxic substance. So can you think of why some bacteria, like the ones causing tuberculosis, they are hard to kill using these mechanisms? So these bacteria generally, they have a defense that protects them from being digested. So it can be oxygen free radical. So an important way our cell kills microbes is by using the reactive oxygen species. So that is known as oxidative killing. So when a phagocytes, it engulfs a microbe, it undergo respiratory burst. So here it consumes a lot of oxygen. So this oxygen is then converted into, that is, super oxide ions by the enzyme that is known as NADPH oxidase. So remember, this super oxide, it is subsequently converted into hydrogen peroxide. So why do you think our cells, they use reactive oxygen species to kill the microbes? So these molecules, they are very reactive and they can damage the cells of the invading microbes, making it easier to destroy them. The other pathway that we discussed was myeloperoxide dependent killing. So we know that myeloperoxidase, it is an enzyme that is found in neutrophils and the monocytes. So the myeloperoxidase, it uses hydrogen peroxide and halide like chloride, bromide or iodide to form hypochlorous acid. So this hydrogen peroxide, myeloperoxidase halide system, it is a antimicrobial mechanism. Other is myeloperoxidase independent killing. Mature macrophages, they don't have myeloperoxidase. They uses hydrogen peroxide to produce the hydroxyl ions and the superoxide oxygen. So they are facilitated by other molecules like iron, etc. So can you think of a reason why the macrophages might not use myeloperoxidase for killing microbes? Well, the macrophages, they have other way to kill microbes, which can be as effective. The other ways could be lysosomal granules. The neutrophils and macrophages, they release granules that contains various enzymes into the phagosomes. So these enzymes along with the reactive oxygen species degrade the engulfed microbes. The enzymes could be protease, alkaline phosphate, and the phospholipase enzyme. Other is the non-oxidative bacterial mechanism that include the lysosomal granules like the hydrolase and cationic proteins like the defensin. Other is nitric oxide. This nitric oxide it is generated by the endothelial cells and the activated macrophages. So why do you think that our immune system have both the oxidative as well as non-oxidative mechanism? So having multiple mechanism, it ensures that our body can effectively respond to various range of pathogens. Apart from the intracellular killing, our immune cells, they can also kill microbes outside the cell. That is by the granules or through antibody mediated lysis or cell mediated cytotoxicity. Can you give an example of immune cells that kills microbes outside the cell? Yes, it could be natural killer cells and certain T cells that are the great example. Now let us discuss about the chemical mediators of inflammation. So these are the chemicals that increase the vascular permeability. That increase the vascular permeability, so they are involved in vasodilation, chemostasis, tissue damage, etc. So why do you think that it's important for our body to have these chemicals? So these mediators, they coordinate with our body response either to any injury or infection. So these what could be the source of these chemical mediators? These mediators can be relieved from different sources like the cells, plasma, or the damaged tissue. So they are broadly classified into two groups: mediators that are released by the cells and they have an origin from the plasma. Now what are the cell-derived mediators? The cell-derived mediator, remember they can be histamine. So histamine, remember it's released from the mast cells. So it will cause vasodilation and increase vascular permeability. Other is 5-hydroxy tryptamine. That is the serotonin. So it is relieved from the platelets and it has a similar effect as that of histamine. Now can you tell where the histamine is commonly found in the body? Well, the histamine it is found in the mast cells and the basophils. The second can be arachidonic acid pathway. That could be cyclooxygenase pathway. That could be prostaglandin. They are again involved in vasodilation, fever and pain. And other is thromboxane. So it promotes the platelet aggregation and vasoconstriction. Other is the lipoxygenase pathway that involves the leukotrienes. So these are. What role do the prostaglandins play in the body? So they are involved in causing fever, pain, and the vasodilation. Third could be lysosomal components that are relieved from the neutrophils, macrophages. They contain enzymes that degrade the cellular debris. The fourth are the platelet activating factors. Fifth could be cytokines like interleukin 1, tumor necrosis factor alpha, interferon gamma. Now, what are the plasma derived mediators? They could be kinin system, that is the bradykinin, that increase the vascular permeability and causes pain. Other is clotting system, it produces thrombin that produces inflammation. The third is the fibrinolytic system that produce plasmin which break down the fibrin clots and promote the inflammation. The fourth could be complement system that produce the complement proteins that enhances the inflammation by increasing the vascular permeability or chemostasis. Now let us discuss about the lysosomal component. For the first is the granules of the neutrophils. We know that they have a primary granules that are known as azurophilic granules. So they contain enzymes like myeloperoxidase that generate oxygen-free radicals. It contains acid hydrolase that helps in phagolysosomes that act within the cells to destroy the bacteria in the phagolysosomes. Other is acid phosphatase that break down the phosphatase. Other is defensine, cathepsin G. What is the role of cathepsin G? It will degrade the proteins. Elastase. What is the role of elastase? It will break down elastin. So why these enzymes in the azurophilic granules, they are so important during the inflammation? So these enzymes helps in breaking down and destroying bacteria so that infection they could be controlled. Other are the secondary granules. They are also known as specific granules. They are alkaline phosphate that is involved in dephosphorylation, lactoferrin that binds iron and inhibiting the bacterial growth, collagenase that will degrade the collagen, lysozyme that will act on bacterial cell wall, plasminogen activator, it converts the plasminogen to plasmin. Apart from that, the third are the tertiary granules. So they contains gelatinase that break down gelatin and collagen, and other is acid hydrolase. So what do you think what will happen if these enzymes, particularly the proteases, they are released excessively? So excessive release, they can cause tissue damage, breaking down the structures like collagen, basement membrane and the cartilage. So in order to prevent the harmful tissue destruction, our body they have a protective mechanism, like they have a anti-proteases, that is alpha 1 antitrypsin They will inhibit the protease activity, alpha 2 macroglobin. Again, they will inactivate the proteases. So monocytes and macrophages in the chronic inflammation also release inflammatory mediators like the acid proteases, collagenase, elastase. So why do you think that monocytes and macrophages, they are more active in chronic inflammation rather than acute inflammation? So we know that monocytes, macrophages, they are the key players in the long-term response to inflammation. So they will help to clean up the debris and promote the tissue repair. Other important is platelet activating factor. So it is a phospholipid mediator that is relieved from the various cells, that is from the mast cells and other leukocytes. They increases. What is the role of this platelet activating factor? So remember that the platelet activating factor, it causes increased vascular permeability, that enhances the ability of blood vessels to allow the passage of fluids and cells into the tissues. It causes vasodilation and vasoconstriction. At a low concentration, it causes vasodilation, that is, it will widen the blood vessel, and higher concentration, it will cause constriction, that is, the narrowing of the blood vessels. Apart from that, it can cause bronchial constriction of the bronchial smooth muscles that lead to narrowing of the airway. So what could be the consequence of this platelet activating factor in a bronchoconstriction patient with asthma? So that would lead to difficulty in breathing and exacerbation of the asthmatic symptoms. Other we have is cytokines. Again, they are the polypeptide substance. They are produced by activated lymphocytes and monocytes. The major cytokine that are involved is interleukin 1, that is produced by macrophages, tumor necrosis factor alpha beta. The alpha is produced by macrophages, remember that, and beta, it is produced by T cells. Interferon gamma, it is produced by T cells. So interleukin alpha and tumor necrosis alpha beta, what they do is they increase the adherence to the leukocyte adherence to the endothelial, induce the release of other cytokines, stimulates fibroblastic proliferation. The interferon gamma, it activates the macrophages and neutrophils. Other are the free radicals, that is oxygen metabolites and nitrous oxide. We already discussed about them. So what is the action of them? They increases vascular permeability, they leads to tissue matrix damage, and the nitric oxide, it is produced by the endothelial cells and activated macrophages. What is the action of nitric oxide? It relaxes the blood vessel, increasing the blood flow. It has anti-platelet activity and microbial action. So what could be the dual role of nitric oxide in inflammation and host defense? So remember that it not only helps in the controlling the blood flow and reducing the clot formation, but it also directly kills the pathogen that are involved in the host defense.